Hey guys! Scientists who would like to reuse the voluminous omics data that are being generated at the moment are typically spending up to 80% of their time to get access to the data, to find out how the data were generated and how the data are represented. To facilitate reuse of data, the FAIR data principles have been launched.
Excuse me.
Hey Junda!
What is FAIR actually?
Well, FAIR, almost everyone knows the acronym by now. It stands for Findable, Accessible, Interoperable and Reusable Data. And everyone is claiming their data to be fair, but actually that's not true in reality because it's quite hard to make data fair, in particular when it comes to omics data. In this short video we will explain how the cross-omics data analysis integration and stewardship pillar facilitates data verification and how this makes the life of a scientist who would like to reuse and integrate data simpler. Let's start with the F, Casper.
Cross-omics launched a FAIR data point that contains descriptions of the cross-omics datasets that we are working with. Both humans and computers can find relevant cross-omics datasets based on these structured and standardized dataset descriptions, also called metadata.
And what about the A, Xiaofeng?
Whether the data are completely open or available on the controlled access, for example, because the data are privacy sensitive, is indicated in a FAIR data point. The FAIR data point may provide access to a FAIR data cube that holds the actual data or points at the location where the data are stored, or to a cloud-based analysis environment where researchers can analyze the data.
Well, interoperability is probably the most difficult part. Anna, what are we doing to make our data interoperable?
We adhere to data standards that are commonly used by the genomics, proteomics and metabolomics communities. And CrossOmics uses the ISA metadata standard, which is short for investigation, study and assay. This allows us to capture metadata on study samples and on omics experiments in a format that is both human and computer readable.
And Purva, how does this actually help the reuse of data?
In the field of bioinformatics, we spend a lot of time mapping identifiers originating from different databases, which is quite time-consuming. So when this data is processed with CrossOmics data verification pipeline, this is not necessary anymore. And there is more to that. The CrossOmics pipeline also provides functions such as data processing and integration that can be run in the cloud or on your local desktop.
This sounds almost too good to be true.
Indeed, we're not there yet, but you can follow our progress on the CrossOmics website or on GitHub.